हाँ आगे से हाँ ओके ओके
टीका मैडम को बोलो कि बस लाइव स्ट्रीमिंग शुरू हो गया है, 1-2 मिनट में स्टार्ट करते हैं।
शुरू हो गया 20 सेकंड में शुरू हो रहा है हाँ हाँ
आप बोलो।
Next, sorry for this. My administrative work is going on, so that's why. So, hello, good morning, one and all. We will be starting in 5 minutes for the scientific deliberation.
Yes, ma'am. We were just waiting for Nishit to stream it live.
Okay.
So he's done now. So everything will be streamed on YouTube, so that the audiences can go through that link as well.
Okay.
I'm audible properly?
Yes, ma'am. रिकॉर्ड कर लेते हैं,
रिकॉर्ड करने से स्टार्ट करिए
पागले से रेडी आप जा रहे हो जैसे और इसका आप करेंगे मतलब बोलने के बाद में कुछ हाँ नहीं यहाँ से कर रही हूँ ना
Listening for the recording.
कर रही हूँ तो जब मैं बोलूँगी कि अब इनकी होने के बाद तो तब आप इसको चला दीजिए
मैं चलाऊंगा वो हमने को बोलिएगा
हाँ बस चलिए मैं आप वहाँ पहुँचकर मुझे सिग्नल दे दीजिए तो मैं स्टार्ट कर बस वहाँ पहुँचकर सिग्नल दे दीजिएगा so we'll be starting in a few minutes. Yes, you can see on your screen it's streaming live on the YouTube.
Yes.
And I'll be recording it as well, so that you can have the records whenever you need it. So I'll be doing that also. Just waiting for a signal from my colleague so that I can start.
Bongade sir is there at your department.
Yes.
Good morning, ma'am.
Yes. Good morning, Suresh.
Good morning.
Good morning, Bagade sir, and all the my budding scientists, researchers from across the globe. Welcome, Dr. Archana madam. Welcome. So we'll start with it.
Yes, ma'am. With your permission, can I please start?
Yes.
Okay. So, this is me, Dr. Archana Mishra. I would like to take you all on a wonderful and fascinating world of the spiders today. So, a warm welcome to everyone present here. And before heading towards the main webinar today, it's my duty and I should pay homage to the, I must say, the super scientist or the first scientist, that is Tathagat Bhagwan Gautam Buddha, and to the great revolutionary stalwart of the era, of all the eras, I must say, Param Puja Dr. Babasaheb Bhimrao Ambedkar. So let us seek their blessings by lighting the candles. Over to you, Dr. DVNS Suresh, sir.
Thank you, ma'am.
Waiting for the prayers, Dr. Suresh. Everything okay, Suresh, sir?
Not connecting.
I can ask him. Suresh, sir, everything okay? ठीक है? क्या होगा? Start.
Internet problem.
I do not know, madam. I think you cannot hear me as well. Dr. DVNS Suresh, you can hear me?
So technical issues.
Yeah. Yes, I have started it at my end.
Bongare sir, can you try from the Samiti room only?
Madam, can you see I have just started?
बस हाँ नहीं स्क्रीन नहीं आ रहा है ये करो ना बंद नहीं स्टॉप शेयरिंग नहीं हो रहा है
Screen is not sharing.
हाँ स्क्रीन शेयर
आपने कुछ देखा?
नहीं मैंने कुछ
Sir,
Can
sir,
we do
आप
from
वहाँ
the
से-
Samiti room?
Yes, yes. उधर से आप बंद कर दीजिए, सुरेश सर. I'm sharing it from here.
Yeah, because we also get the information about the lightning of lamp that it comes on our screen.
Is it visible, madam?
अभी आ रहा
Yeah.
है इसको बंद करो शेयरिंग
It's not sharing.
Yeah, once.
वो मैडम कर सकते हैं वहाँ से बंद हाँ
बंद करूं?
हाँ कर दो ना शेयरिंग स्टॉप शेयरिंग करो अभी ठीक है मैम, आप अभी आप दिख रहे हैं।
हाँ, मैं शेयर कर लूँ?
हाँ, आप स्टार्ट करो।
Is it visible? Is it visible, madam?
No.
Okay, because I can see it here. Anyways, I think then we should proceed.
Why that clip is not going on of lighting the lamp?
Yeah, but to me it's visible over here. When I'm sharing it, it's visible to me.
It's not visible to me. You share, see when the guest can see it.
No, I don't think so. Anyways, in that case, I guess we should.
मैम अभी आप दिख रहे हैं,
हाँ
प्रिंसिपल मैम आप अभी दिख रहे हैं।
हाँ मैं दिख रही हूँ, पर वो जो स्क्रीन जो अपने को करना था वो नहीं दिख रहा है ना।
ये इनका बंद हो गया, इनका पीसी है ना, इनका बंद हो गया है।
सर आप
Hang हो
समिति
गया।
रूम से कर
Hang हो
सकते
गया।
हैं।
सर आप
Yes.
समिति
Hang हो
रूम में
गया।
आ जाइए।
मैं-
घुंगारी
हाँ,
सर, आप समिति रूम में आ जाइए।
मैं आ जाता हूँ।
Suresh sir, I am not the host. That is why maybe I'm not able to share the screen.
Okay.
So if at all you guys
To make Archana ma'am the host, co-host.
Yeah, ma'am. I'm still
The PC is not working of the zoology department.
No, it was working because of the internet. It has got stuck.
Okay.
I have restarted it.
Yeah, Bongade sir will come to the Samiti room and
He's there with me.
share the slide.
Yeah, shall I share it again because it wasn't shared that time?
Yeah.
Please.
Yeah, I'm sharing it again.
Okay.
Madam,
I'm so sorry for the inconvenience caused due to the technical problems.
Yes.
Archana madam, please make me co-host.
Yes, sir. Sure. Yes, proceed, Dr. Devanus.
I think it's sharing now.
Yes.
Yeah. Yes.
Okay.
So these are the blessings from our Lord Eternal Tathagat Gautam Buddha and Param Puja Dr. Baba Sahib Ambedkar. Blessings are always with us. Internet problem is there something.
After seeking blessings, am I audible, madam?
Yes, you're audible.
Yes. After seeking blessings from Tathagat Gautam Buddha and Parameshwar Dr. Baba Sahib Ambedkar, I once again welcome you all to this wonderful journey of spiders. I respectfully bow down to the rock solid pillars of our management, our patron, Honorable Madanta Arya Nagarjun Suresh Society, the President of Param Pujya Dr. Babasaheb Ambedkar Smarak Samiti Diksha Bhumi Nagpur. And sort of words to welcome and thank our co-patron, Dr. Sudhir S. Fulgere sir, the Secretary of our Param Pujya Dr. Babasaheb Bhimrao Ambedkar Smarak Samiti and Director of Management Research Studies, Nagpur. Of course, no words can describe the determination and encouragement we receive from our motherly principal, our chairperson of today's webinar, Dr. Mrs. Bhuvneshwari Mehre, Madam. Thank you so much for being there, Madam. And it gives me really pleasure to work with worthy colleagues like the convener of today's webinar and our head of zoology department, Dr. R.S. Bagre, uh, uh, big brother, I must say, who is always there to support me. And again, one of my worthy colleague who is the co-convener of today's webinar, Dr. DVNS Suresh. He is a wonderful colleague, I must say. So it gives me really pleasure to welcome you all on this platform of Zoology Department of Dr. Ambedkar College. So before marching and heading towards our main webinar today, I would request the principal and chairperson of this webinar, Dr. Bhuvneshwari Mehre, Madam, to please bless us by her motivating words.
Thank you, Archana. Thank you. Thank you for your words. Hello, a very warm and pleasant good morning to one and all. At the outset, I, Dr. B.A. Mere, Principal of Dr. Ambedkar College, welcomes you all for today's international webinar organized by Department of Zoology on the topic Diversity of Spiders and Geologic Evolution. As we are very aware of a very difficult and dreadful situation of pandemic COVID-19 all over the globe, and hence we are organizing all the educational events and extracurricular events on Zoom platform and YouTube. So at the outset, to initiate, we first seek the blessings of two stalwarts of humanity, Lord Gautam Buddha and revolutionary legend, architect of Constitution, Dr. Babasaheb Ambedkar, whose blessings they are always with us. Today's topic of deliberation is basically for researchers and innovators. The air-breathing arthropods and ranked 7th in the total species diversity among all other groups of organisms. They are ancient animals with a significant role to play in ecology and maintain the ecological equilibrium. Spider silk, an alternative to silkworm, silk fiber, knowledge on molecular properties and different insight will be taught on the full open insight will be interactive sessions will be a major aim. So we are very fortunate today to have with us from across the globe young budding scientists and researchers. Today's guest, Darko from Humboldt, postdoc fellow, Senckenberg, Academy of Sciences, California. I welcome you, Darko sir. Welcome at Ambedkar College. Another guest speaker, Rashid Wongprong from Thailand, nature educator, fellow and director of Thai Nature Education Center. I once again welcome you, sir. Rashid sir, welcome at Dr. Ambedkar College. The other research fellows, Joseph, I have seen you, Joseph is there. So welcome at Ambedkar College. The convener of the program, Dr. R.S. Bagre sir, head of the department Zoology, the convener of the program, co-convener, assistant professor from Zoology department, DVNS Suresh sir, and my colleague and one of a very most important instrumental, my colleague Archana Meshram madam, Dr. Archana Meshram. She is instrumental throughout this organization of this webinar. I welcome you, madam, and a very warm welcome to you for this organizing such a international webinar from spiders. My worthy colleagues, Dr. Revatkar, Vice Principal and Head Department of Physics, IQAC Coordinator, Dr. Hema Menon from Law Department, all my dear students who are streaming online on YouTube and on Zoom link. So I once again welcome you all for spider silk and most incredible hierarchical order materials which possess outstanding properties in this world of spiders. Today's scientific deliberation will give an complete insight on the greatest knowledge, and the biological scientists across the globe will justify the topic, and my students' urges will be fulfilled through this deliberation. Sir, our college, which is established in the year 1964 by late Padam Shri Dada Saheb Gaikwad, and with the two luminarians, Dr. Honorable Shri R.S. Gavai Sahib, President Smarak Samiti, and a driving force and fatherly figure, Mr. late Shri Sadanandji Fuljale Sahib, who has at the forefront to start or initiate this institution at a holy sprawling land of Diksha Bhumi, Nagpur, where Dr. Baba Sahib Ambedkar embraced Buddhism in 1956. Sir, this land is land for transformation. As an educational institute, here we are transforming the mind of the young ones, young budding students, to explore the world for the benefit of the mankind. So I once again welcome you, sir. So the exponential growth of this college is seen under the leadership and blessings of present President, Honorable Bhadan Arya Nagarjun Surai Sasai, and young dynamic visionary secretary and director of Institute of Management and Studies Research, Dr. Sudhir Fuljile sir, at constant and dynamic role with dedicated academicians, technical staff, administrative staff, and their hard work, meticulous planning, execution, fulfill the mission and vision made to initiate the most trusted center for quality education in this India, or basically the central Nagpur, and that is India. So I once again welcome you all for today's scientific deliberation, and I know that you will justify with the students, you tell or interact, whatever the research is going on, you will deal with them, you will interact with the student, and students will satisfy, you give a satisfied answers, so my students will get insight on this new novel research about spiders. So I once again welcome you all from across the globe, and thank you all, and congratulate once again all the organizers, especially the team from the zoology department, Dr. Bagre sir, Dr. Suresh sir, and Dr. Archana Meshram madam. Welcome you all, and thank you. Thank you all.
Thank you so much, ma'am, for the inspiring words. As we all know that there are more than 45,000 known species for the spiders and found in all the habitats all over the world.
Okay, sorry.
May I ask everyone to please mute themselves? Okay. The spiders, or I must say the araneae, they are air-breathing arthropods and which have 8 legs and chelicerae with fangs that inject the venom. We all know this. And they are the largest order of the arachnids and rank 7th in the total species diversity among all other groups of organisms. As we all know that spiders are ancient animals and they have a history which goes back to 350 million years ago. They are abundant and widespread in almost all ecosystems, and no doubt they constitute one of the most important components of the global biodiversity. Apart from that, they have a very significant role to play to maintain this ecological equilibrium and the ecology, because they are exclusively predatory animals. And the participants today, they will be introduced through this webinar to the biology of the spiders, and with the emphasis on the ecological role of the spider especially, which my friend Darko will speak on, and the incredible diversity, Prasit will take care of it, and the techniques to identify the specimens to the species level. Other topics which will be included here today will be the evolutionary relationship, functional morphology, behavior, and physiology of the spiders. So I must say, of course, they are very fascinating animals. I always found them fascinating because of the 8 eyes, the eye arrangement. Actually, it differs from species to species. And then the 8 legs, the venom. And it's a perfect combination of beauty, poison, predator, wonderful eye arrangement. And not to forget about the silk, which is really a constituent of protein, and it possesses large qualities of non-polar and hydrophobic amino acids like glycine and aniline. And it is most of the versatile materials on the earth which we can find. So actually a protein which is created by the special organs, they are known as spinnerets and all. So today's speakers will take you into a deep study of all these things. So I will not be speaking much on it. And the best part I love about the spiders is that, in spite of being very colorful, very beautiful, with this unique eye arrangement, with the venom and everything, people actually get scared of it. But researchers have found that they too have feelings. They too snuggle their young ones and they too take care. Parental care is there, neoteny is there. So that makes it a very most, I must say, wonderful creatures. And my respect goes to Dr. Ganesh, late Dr. Ganesh Wankhede sir. He was truly the Spider-Man of India who has introduced me to this wonderful field. And I was like doing a DSc under him, but unfortunately we lost him a few years back because of blood cancer. So today I would like to pay a tribute to him as well. And for the further proceedings, I would like to call upon my HOD of Zoology Department and the co-convener of today's webinar to introduce you all to our Zoology Department of Dr. Ambedkar College, Diksha. Over to you, Bagre sir.
Hello, good morning. Hello, good morning, everyone. You are welcome for this good wonderful event. It's a kind blessing for Tathagata Bhagwan, Professor Buddha, and Bharat Ratna Dr. Baba Saheb Ambedkar. Zoology Department organized this wonderful event today on the biodiversity of the spider. I, Dr. R.S. Bagre, the head of Department of Zoology. We are here three members of the teaching staff. Dr. V.A. Sure, Assistant Professor, Dr. Ratna Mishra, Assistant Professor, and three are the technical staff. With the cooperation of our staff, every year continuously and consistently organized each and various activities for the progress, achievement of the students with respect to the academic research in various applied fields which are useful and to the society. Every year, our department trains the students with the nature club. Dr. Archana Mishra is the coordinator of the nature club. Nature club organizes various workshops, various lectures, various activities regarding the maintain the biodiversity as well as multiplication of the nature aspects, which are very, very useful to the society. Department have ECGC cell. Dr. Suresh is the coordinator. Through these cells, the students is able to encourage, enhance towards the competitive exam, placement, certain different job purposes in different fields and so. Being the convener of this international webinar on diversity of the spiders and geologic evolution, first of all, hearty welcome and good morning to the day. Number one is that Darko D. Kokar, Chandan, Postdoctor, Fellow, Edinburgh Museum Research and Associate, California Academy of Sciences. Second year friend is the Prashit Wangko, nature educator of the Thailand. He accepted our invitation for this webinar. And also, hearty welcome to all the participants, students, academicians, research throughout the world. It's the first time to organize such a kind of wonderful webinar on a spider because it's a new field in the arthropod for the academicians, research, and educators as well. Spiders are found everywhere, from blazing desert to freezing plains, exhibiting extreme resilience, adaptability, and unique survival mechanisms. Broadly, spiders are divided into two categories. Each spider, these are also called as Ligoloma, and a small spider, which are the ANU Mars. India is a home to several unique species of spiders. Some are known to the science, while many others are yet to be explored. According to an estimate, India is a home to 59 families of the spiders. Although spiders are often discussed based on their regional distribution, such as South Indian spider, and so on. They are not bounded by this territory and can be found all over. Despite their widespread presence, spiders are poorly studied. Very few known records of their natural history exist, not only of those found in the wilderness, but of many commonly seen spiders in homes, parks, backyards. Till today, most descriptions are based on the preserved specimens. Field study based on the living specimen collected and observed from the field enable easy identification. There is a need to record their behavioral patterns, natural history. Spiders spin webs using thin threads of the silkworm. Their spinnerets, these are comparable to kevlar in strength. Webs play different role like besides to trade or to capture the prey. On this occasion, I appreciate and congratulate the organizing secretary, Dr. Archana Mishra Madam, and coordinator, co-convener Dr. D.V.M. Suresh, to chosen a wonderful event for the creativity of the students, researchers, and educators as well. I hope it will be grand success. At the last, on the behalf of my zoology department, I am very thankful to Patron Honorable Bhante Aryan Nagarjun Surai Safai, President Param Pujya Baba Saheb Smarak Committee, Holy Dikshavumi, Nagpur, Maharashtra, India. To patron Dr. Sudhir S. Kunjale, Secretary, Param Pujya Baba Saheb Ambedkar Smarak Committee, Dikshavumi, Nagpur. Chairperson Dr. Mrs. B.A. Meher Madam, Principal of the college. Time to time to support, cooperation, and availability of the facilities to organize the wonderful event. Thank you very much.
Thank you, sir. That was indeed a brief introduction of the zoology department of Dr. Ambedkar College. Friends, it's said that science knows no country because knowledge belongs to humanity. And it is the torch which illuminates the world. So it is not that we use technology, but in this era, and especially in this pandemic, we are living technology. So actually, we must thank technology that in spite of all the odds on this one platform of Zoom, we could connect with people from various countries over here, and we could have a brief introduction and we could get acquainted to know each other. So let me have the pleasure and honor of introducing my first speaker today, Dr. Darkodi Kotoras. Dr. Darkodi Kotoras is a Humboldt postdoctoral fellow at Senckenberg Museum and is working as a research associate with the California Academy of Sciences. His main research interest is on the historical processes that create biodiversity in the condition of isolation. He focuses especially on invertebrates from islands in the Pacific Ocean, and previously he did a postdoc between the California Academy of Sciences and the University of California, Santa Cruz, on improving ancient DNA methods for museum specimens. He is sponsored by a Fulbright Fellowship and he earned a PhD on the Integrative Biology Department at the University of California, Berkeley. Here he studied the temporal dynamics of Tetragnatha, which are the spiders, they adaptive radiations in the Hawaii. Before he got a master's in ecology and evolution at the University of Chile, working on Evo on the same institution, he did a bachelor's in his biology. So I'm very blessed, I must say, and thank you so much, Darko, for the friendly gesture you showed. that within seconds you accepted my invitation to present your work and introduce our students and the people of India to your wonderful research. So can we please have your, like it's really being late and we are very desperate to see your presentation. So over to you, Darko. You can please share the screen as well and welcome once again.
Well, I would like to start saying thank you very much to you for the invitation and to everyone attending for your interest on the work that I'm going to present. I'm very happy to be here virtually, and I hope there will be opportunities to meet in person, in reality, once COVID is gone, hopefully.
And Darko, we are streaming this on YouTube as well live. So we need not worry about the participants on the Zoom link because you are being heard here now through the live through the YouTube channels.
Excellent. Okay, so without further ado, I'm gonna start telling you about my work. Today, what I would like to do is to tell you about how the geologic evolution of a volcanic archipelago, the Hawaiian Islands, shape the biodiversity of the species living over there. In particular, a group called the Tetragnathus spiders. The talk has mainly two big parts. The first one, it's an introduction to the geologic history of the Hawaiian Islands. So that will be more like some sort of basic geologic class about volcanic islands. And then in the second half, I will present original research about how using population genetics and interconnecting that with the geologic history, we could understand why some groups are so diverse in this volcanic archipelago. So let me start with the general conceptual framework of this work, which is related with adaptive radiations. I think adaptive radiations are perhaps one of the most interesting phenomena in evolution, and they correspond to the rapid diversification of a large number of species in a very short period of time. Associated with that diversification, we have the origin, we have some ecological trait, ecological axis, which allowed the species colonization of different micro niches. So adaptive radiations happen at different timescales. We have them at millions of years, like the Australian marsupials. We have them in hundreds of thousands of years, like the East African cichlids, and in couple millions of years, like what we see in many volcanic islands, such as Hawaii, the Galapagos, etc. They also happen at very different types of geographies. We have them in real volcanic islands, continental islands, lakes, mountaintops, etc. They tend to occur in isolated places. And finally, adaptive radiations are very common throughout the tree of life. We find them in vertebrates, invertebrates, plants, etc. So because of all these reasons, looking at this phenomenon has a lot of generality, and understanding its mechanisms in one study group could inform us to what's happening in other groups of organisms. Now, besides the fact that we know a lot about this phenomenon, there are some key points that are still missing. In particular, understanding how does mechanistically this explosion of diversity, this explosion of concerted speciation events happen at the very early stages. And that's what we're going to try to explore, what we will explore today. So to kind of give you an idea of which tools we're going to use to study this, I would like to invite you to think a little bit outside the box and forget about spiders and islands for 1 second, and imagine that you are the Minister of Education of a country. And then it will become clear why I'm asking to do this. So imagine you're a Minister of Education and you want to see what's the effect of some type of educational reform. And then there are many ways where you can see the effect of educational reform. One of them would be to give them a test to the students. So you implement the reform, you give them a test, see how they do when they are in first grade, then in second grade, third grade, and then once they graduate, you go and talk to them. Then you can gather more information than how they they perform in mathematics. You can learn about their knowledge in history, art, etc. So this, what I just described, is very similar to what paleontologists do to understand biodiversity dynamics. They will look at the fossils, which are kind of the exams, and they will look at current species and being able to gather more information. This paleontological approach, this diachronic approach, allow us to track diversity changes through time. Now, there is a problem that when you look at volcanic islands, many of the fossils, the conditions of fossilization are not ideal. So true fossils, like what you think normally, are not present on volcanic islands. They are mostly subfossil remains. So in those cases, there is another approach, which is the synchronic approach, where you implement this reform, and instead of looking the same cohort of students through time, you can go and interview students at different ages, first grade, second grade, and so on. If you're asking something that is related with a very stereotypical process, something that repeats over and over again, despite the individuals going through this process, for example, the educational system, it really doesn't matter that you're tracking the same person through time. You can look at different cohorts and still gather a lot of information. This is a synchronic approach, and in geology has been called the chronosequence approach. Basically, you look at different places, you look at the same time places that are evolving at different moments. And this is what has been studied extensively in volcanic islands, particularly in the Hawaiian archipelago. This tropical group of islands are located in the tropical North Pacific between South America and Asia. They start with the tallest islands, then they extend towards the northwestern Hawaiian Islands, and further on, they become a long underwater mountain chain that finally subsides under Kamchatka in Russia. This is a close-in where you can see the tall Hawaiian Islands, and then the northwestern Hawaiian Islands, which are a collection of rocks, atolls, and shallow reefs. All these islands at some point were down the southwest, where is currently the hotspot, the volcanic hotspot. If we zoom in even more, we see the tall Hawaiian Islands, and these are the ones that we're going to be focused on. The Big Island, which is the youngest, the Maui Nui complex with Maui, Molokai, Lanai, Oahu, and Kauai. So now let me quickly tell you about what is the life of a volcanic island. It all starts under the ocean, where volcanic activity starts building up these shield volcanoes, and eventually they pop up from the sea level. And this, the best example for that is the Big Island. And here I'm going to refer to a few geologic features that are important. First is the fact that a young volcanic island is actively growing, and that's the case the Big Island, of course. At this stage, the island is expanding its area constantly and producing new land which is very poor in terms of soil. All the minerals are still captured within the rock. We can see that the type of lava produced in a volcanic hotspot is different to the one that you will find in a subduction volcano, such as the ones that you find along the Andes. This lava in the volcanic island, it's more viscous, tend to be rich in iron, and comes from deeper magmatic chambers. One interesting phenomenon that occurs here is that sometimes lava flows will go through existing forests, creating, in geologically speaking, 1 second or immediately some sort of volcanic systems, some sort of island systems, islands of forest, which favor the fragmentations of populations. This it's a picture of a lava flow from 1835 that fragmented all these pockets of vegetation in effectively a blink of an eye, geologically speaking. Here you can see how the matrix in between these pockets of forest, it's inhospitable habitat that prevents migration between the species living here. This situation has been studied as a potential driving force, as a potential pump of a species diversity, favoring these transitory events of isolation and later admixture. Another element important to describe on the Big Island is its massive size. It reaches in Mauna Kea 4,300 meters, having snow during the winter, like right in the tropics. So this high elevation provides provides an altitudinal gradient where you can imagine different ecosystems occurring with elevation. So that's another reason why diversity is so high on these islands. Another interesting fact is another interesting element is the fact that the islands start geologically very simple. Sheer volcano has smooth slopes, but with time, erosion, landslides, and other processes, which I explain later, increase the topographic complexity. So as the island shrinks, the topographic complexity increase. And here we see different, we see different lava flows that representing the ages of the 5 main Hawaiian volcanoes: Kohala, Mauna Kea, Mauna Loa, Hualalai, and Kilauea. We can see that there is some sort of progression, but there are also some areas with really old lava flows surrounded by young lava flows, like this area on the southeast corner. Now we move on to Middle Age islands in the Maui Nui complex. Here we have the island of Maui with Haleakala, 0.8 million years, and West Maui, 0.2 million years. On this island, we can see how at high elevation, it's an alpine environment, 3,800 meters, extremely dry, and clearly a volcanic area with all these cinder cones. Another element that we can see here is the presence of 2 very big valleys, very big gaps, which is not very well understood how they were produced, but also add a little bit of the complexity that I mentioned, that as the island becomes older, it appears. The Molokai, which is another island, has a very curious shape, like a rectangle. And here you can see how there are all these gullies towards the south, but they're absent in the north. The reason for that is that the north has very steep cliffs, more than 300 meters cliffs, which are the result of a massive event of landslide, where literally half of the island washed off to the ocean. And then it created this very strange shape. Another thing important to mention on these Middle Age islands is that the soil quality is already increasing because of the vegetal matter. Nitrogen goes up because of weathering. There is a release of different minerals like calcium, and also the island itself, because of its weight, starts deflecting the bottom of the ocean and the island starts sinking. So now, as the island gets older, it starts losing elevation as well as losing some of its matter due to landslides. Lanai, it's another island around the same age. It's drier because it's in the rain shadow of these 2 volcanoes, the same as Kaho'olawe, which is even lower in elevation. Very similar to what I described before with the Kipuka dynamic, this fragmentation due to lava flows. We have something similar occurring related with the subsidence of the islands. So here we have Maui Nui complex today, where we have these 4 islands. But if we go back in time 1.2 million years ago, we will see that it was a more massive island with all these volcanoes connected between each other, as well as areas that today are shallow waters. As the island becomes older, it starts sinking, and this one single island turns into a little archipelago. Interspersed in between this process of sinking, we have glacial periods during the Pleistocene era, which changes the level of the ocean, allowing for short periods of connection during the interglaciers of these islands that are, during the glacial periods of these islands that are being separated because of the subsidence. So here we have another very similar dynamic of admixtures and isolation, but in this case happened in the temporal scale of hundreds of thousands of years. Oahu, moving even further north, it's another island with 2 main mountain chains, the Koholau Mountains and the Waianae. In the Koholau, we can see very clearly the effect of rain erosion, creating these shaped blade mountain chains that are the result of water erosion. Hawaii, it's a very, very wet place. Some of the areas like Mount Waialeale or Kukukui reach up to 7 or 8 meters of rain per year, which is amongst the highest. The wettest place on Earth. It's in India. I don't remember the name, but it receives around 10 meters of rain, of monsoonal rain. In parallel to the erosion of the islands, we have the build-up of coral reefs. This is a tropical island, and corals create what in the end will become the ring that forms the atoll. Here we have some type of fringe coral on the Hanauma Bay, which is a shallow ancient volcano crater. And on the edge, we see a little bit of coral development. Here in Hawaii, it's less than other islands in the South Pacific because of the temperature as well as the steep profile of the island. Volcanically speaking, there is another feature to mention on these older islands, which is the rejuvenation process. Rejuvenation corresponds to the creation of new volcanic craters due to volcanic chambers that got kind of trapped once the island was at the hotspot area, and then later in the life result in very distinct and punctual volcanic events. One example is Diamond Head, which is this famous picture of Waikiki, taken from Waikiki, and then Coco Head on the back. Finally, to introduce the geology of these islands, I'm going to talk about Kauai, the oldest of the main Hawaiian islands with 5 million years. One interesting feature is this little mountain chain called this little mountain chain here. This mountain chain, very small, very little, is actually one of the oldest mountain chains on the volcano. And that reveals how sometimes the surrounding areas could be eroded and leave these very old lands. This mountain chain, the Haupu mountain chain, is so old that it has developed endemic species from a group of ligniphids called the Orson whales, an endemic genera from Hawaii. Similar in principle looks the Napali coast, where we see these mountains going right into the ocean. But the geologic mechanism is different. These are not particularly old. However, they have this steep profile due to the wave erosion that is produced by swells coming from the north. If anyone's like surfing will know that most of the prime surf spots in Hawaii are located in the North Shore, in Oahu or Peahi in Maui. In this case, it's the same thing. During the winter swells, big wave action will erode the land. Also related with erosion, we have the spectacular Waimea Canyon in the center of the island, which is the result of a process where the island first grew, and then half of it was eroded or kind of not washed away, but it disappeared due to landslides, and then was refilled over 1 million of years, kind of creating the other half again. These two pieces of the volcano with 1 million years of separation did not really fuse. And then with time, they separated again, creating this massive canyon. In this volcano, Waialeale, it's one of the highest wetlands in the world with 1,400 meters and corresponds to the Alakai Swamp. Okay, so that's kind of the overview of what happened with the geology of a volcanic island. Now, another important element to understand the diversity of habitats in Hawaii is also the rain patterns. I already talked about elevational gradients, but if we see rain gradients, they're also as extreme. Hawaii is in the path of the trade winds from the north. So we can see here in cold colors, the mean annual rain precipitation in millimeters. And we can find areas with between 7 and 10,000 millimeters on this western coast, on this eastern coast, and then areas with less than 300 on these western areas. All of this, please note that this in less than 50 kilometers. So in less than 50 kilometers, we find a gradient of more than 7 meters of rain. So that coupled with the elevation can give you an idea of habitats and ecosystems can get compressed or are compressed in this archipelago. Now, today, I'm going to talk about a group of spiders that is really focused, that is really present in the montane rainforest, which looks kind of like this. Interestingly, in Hawaii, as a good isolated island, taxonomically speaking, at high levels, like genera of genus or family, is not very diverse. If you know the families and genus from one island, you will know pretty much all the families and genus from the other islands. But at the species level, it's very diverse. So each island, in some cases, many volcanoes will have species endemics. Another element that I already mentioned is the fact that the islands are constantly subsiding. So that creates some sort of some sort of losing of high elevation environments with time. Here we have Mauna Kea and Mauna Loa, 0.01 and 0.3 million years old, going up a couple thousand meters above the cloud line. So here we find alpine environment. Now in Haleakala with 0.8 million years, the amount of alpine environment is reduced. When we see West Maui, 1.3 million years, there's no dry alpine environment. The top of the island, it's completely covered with mountain rainforest. And in the Ko'olau, with 2.6 million years in Oahu, further north, we see that the inversion layer kind of touches the very top of the islands. So this gives us an idea of how the habitats, especially high elevation habitats, are shrinking with time. And finally, this cartoon shows us the whole archipelago, and an interesting element to highlight is the distance between the islands. Between Oahu and Hawaii, we have about 300 kilometers, but between Kauai and Oahu, 150 kilometers. So the frequency how close you find the islands, it's also a reflection of how active was the hotspot and how quickly was moving the plate. So both things in parallel are kind of shaping the distribution of these territories. So all of what I described before is the scenario, is the landscape that produces the biological diversity that we find today in Hawaii, which to my opinion is one of the most spectacular that we can find in the world. And given these conditions, we can also understand what are the geologic drivers that produce it. So looking now at the species, Rosemary Gillespie and Bruce Baldwin created this plot, this very insightful plot, where they plot the number of endemic species standardized by area against the age of the island for animals and plants. And what we see is that the groups with more number of species, the big radiations, overshoot in standardized diversity on middle-aged islands, and then they decline towards Kauai, while smaller groups kind of increase constantly towards the oldest island, which you can consider some sort of neutral model. The oldest the place, the more species it has. Now, the fact that in a middle-aged island there's a peak on diversity, that could be telling us something about the process of adaptive radiation itself in terms of its temporal dynamic. And here we can use the chronosequence approach to understand mechanistically how does it happen. So let me present this model, this conceptual model of how we think we can understand this overshooting diversity on middle-aged islands. Let's imagine we have a young island with a patch of rainforest and one species. This volcanic island creates a new volcano. The species colonize this new volcano. Lava flow process creates this dynamic of admixture and isolation, and then the process repeats for a third and a fourth volcano. Then we end up with 4 isolated populations of the same species. As the island gets old, these 4 populations could turn into 4 allopathic species. And as the island gets even older, some of these areas could disappear due to the processes that I described, the geologic processes I described before. So then we have a decline on diversity. We go from 1 species to 4 to 2. Now, more complex things could happen, like secondary contact of the species, which allows survival after the extinction of the original area of origin for the species. And with secondary contact, there could be hybridization. So this could become as complex as you want. And this is kind of the frame that I will try to test with real data. If we look at spider species now, we'll see there are two main modes of speciation. One of them, it's non-adaptive radiation, which is illustrated by the Orson Welles spiders, basically several concerted events of allopatry. And that's where we see here, basically each volcano or island will host its own species. They're all ecologically very similar. On the other end, we have adaptive radiations, like the example of the Ariadnes. These spiders evolve with geology, just like the Orson Welles. But once they colonize one volcano, they will diversify across an ecological axis. And that is represented here with the colors on the phylogeny. So we not only have the non-adaptive radiation element due to isolation, but also ecological differentiation. The same thing happened with the group of study for this presentation, which are the Tetanata spiders. This genus, which has a worldwide distribution, hosts 11% of the world distribution in these tiny islands. That gives you an idea of how diverse this group is here. They are the result of actually two adaptive radiations. One, the spiny leg clade with 11 species. These spiders are active hunters. That's something that no other Tetanata do anywhere else. And then we have the web building clade, which is a group that spin webs and diversify along that ecological axis. The spiny leg clades, they also have evolved 4 different ecomorphologies. An ecomorphology is a collection of ecological syndromes that relate to the actual ecological niche that species is using. In this case, we have the green spiders living under leaves, the maroon living in moss, the big brown living in bark, and the small brown or little brown living on twigs. We found these 4 ecomorphs occurring independently on all the islands. And that's represented here by the pie charts, the ecomorph and the name, what's a species. As a well-behaved adaptive radiation, you don't find 2 species from the same adaptive radiation, from the same ecomorphology living together. The same way, like metaphorically speaking, on a town, you will find 1 mechanic and 1 baker. You don't find 2 people doing the same job. Now, on the island of Maui in Haleakala, this rule seems to be bended a little bit, where we find 3 species of the green ecomorph occurring on different areas of the volcano. So this is interesting because this could give us an idea, or by looking at the situation closely, we can see this moment where we overshoot in diversity. And here are the species that I'm going to be looking. They all have populations on Haleakala, but they also have other populations in another volcano or island. The species are Brevignatha, Waikemoe, and Macrocantha. So we could hypothesize how this situation was created and try to link with the proposed model that I showed before. We can think that originally there was 1 ancestral species with 3 different populations, and by allopatry turn into 3 different species, and then with time move around and reach the current distribution. Another hypothesis is that this ancestral species was distributed exclusively in Haleakala, and due to parapatric speciation turned into 3 species that later on distributed around. Now, these 2 hypotheses are the extremes. You can imagine all the scenarios in between, where, for example, 2 species were produced by allopatry, 2 species by parapatric speciation and 1 by allopatry. Also, probably all the species move around, but only 1 species got settled in these other volcanoes. So to test all of that, what we could use is population genetics and understanding which of the 2 scenarios it's the one that is favored. We can test if our proposed model has data that supports it. I'm not going to go into the technical details. I will be very happy to answer them later. But basically, what we did was exon capture, which is a mechanism, which is a method that used next generation sequencing to capture several thousands of genetic markers from independent areas across the nuclear genome. We design a capture rate based on a transcriptome. Here are some numbers. The target size was 1.7 megabases, representing 1,800 ORFs. I don't talk about genes because we did not annotate them. We multiplexed this hybridization experiment, and actually 3 independent experiments. We were able to put everything together because we independently barcode each of the specimens. After running the hybridization experiment, we ran separate tests to make sure that the hybridization worked, positive controls, negative controls, as well as looking at fragment size distribution. Once all these controls were successfully, once we checked that they successfully resolved, we sent this to sequencing to a Illumina platform, each experiment on a different run of Illumina HiSeq 2000 back then. This is a few years ago already. And once we got the data back, we did the processing following the Museum of Vertebrate Zoology pipeline at UC Berkeley. Here are different programs. I'm not going to talk about them in detail, but basically, once we get the raw data, we clean it, then we assemble the raw reads using our target as a first reference. And after doing creating one first, after mapping everything to the target, we create a new reference. And against that new reference, we remap individually each of the libraries. After doing all that mapping, we then create VCF files and use ANGS, which is a program that uses side frequency spectrums as a way to then run more common population genetic analysis, such as principal component analysis, phylogenetic networks, admixture analysis, etc. So the power of this analysis was to use side frequency spectrums instead of calling directly SNPs, because that allows us to use data which has lower coverage. So yeah, these are the side frequency spectrums, and these are the post assembly analysis. Okay, so that was the technical part. Again, I will be happy to answer more detailed questions at the end. And let's go back to the biology. Remember, we have this species, and we wanted to understand how they reach their current distribution. The first result is a principal component analysis. Each point represents one specimen, and the colors represents the species identity based on morphology. Now, spiders spend most of their lives as juveniles, and juveniles in this case are impossible to tell apart. So here in this plot, you will see with color those individuals that were morphologically identified. In gray, the ones that are juveniles, and we couldn't tell based on morphology, but the shape will tell us about the potential identity due to their location. Remember that each species occurs in a separate locality. So by looking at this, we have the first surprise. We have 3 species, but we have 4 clusters. And the one that they get split is Tetranatha brevignatha, which has populations in Maui and the Big Island. So we can comfortably say, based on genetics and comparing with the other species, that we are in front of a cryptic species, Brevignatha from Maui and Brevignatha from the Big Island. If we look at close, these are the specimens from the Big Island, and we find green circles and gray circles. So adults and juveniles. The same story here in Maui. Now, if we look at the cluster over here, it's orange, representing Tetranatha macracantha, and we find, for example, an orange square. So it's macracantha based in morphology, but also based in locality. We have gray squares, so these are juveniles, and they cluster genetically with the adults, so that's fine. We also have this diamond, which corresponds to new records, areas that we didn't have specimens before, and morphologically and genetically, they end up being also from the same species. So nothing very spectacular, nothing very unusual here. Perhaps interesting to point out that despite the fact that the species is present on 2 islands, genetically it appears as one cluster, which is different to what we saw before. When things get really interesting is when we look at Tetranatha waikamoe. Here it's blue triangles, what we expect to see. We find blue triangles, we find gray triangles, excellent. And we also find blue squares. Blue square means that morphologically it was an adult of waikamoe, but it was placed on a location where we expected to find macracantha. We also find gray circles. So it is a juvenile that was found where we expected to find brevignatha, but clustered genetically with waikamoe. If we zoom in on this group, we will see that the two volcano populations of Waikamo split, and it is in East Maui, in the volcano where we actually saw the three species occurring in different areas, where we find this situation. So the main finding here is that Ternata waikamoi actually co-occurs with the other two species. There is sympatric distribution, and this was unknown before. Now, because there's sympatric distribution, we can then ask, is there hybridization? And for that, we use this admixture analysis, and looking at closely the two populations where we find this co-occurrence, particularly Kipahulu Valley and Lower Waikamoi, we find that there's no signal of hybridization, and that is represented by these solid lines. Each line represents an individual, and the colors, the genetic background. If it's solid, one color means that it has only one genetic background and not a combination like here. But for the two populations, we don't see hybridization. Then this result is further confirmed with a phylogenetic network by actually use calling SNPs. Instead of using side frequency spectrum, we call the SNPs. So same raw data, but different type of data analysis, and we find the same result. All the individuals of Ternata waikamoi present on East Maui clustered together, and they don't show up in in-between position, which is what you will expect if they are actually true hybrids. I would like to also point out that this analysis actually allow us to separate the two populations of Macracantha. Remember this group present here, that the principal component analysis show it as one single cluster. This other analysis is more refined, if you want, and allow us to separate Lanai and Maui populations. Yet with this stronger power to discriminate, it's not showing any degree of hybridization here. So, and now to start wrapping up, what have we learned? First, the three nominal species cluster in 4 or 5 clearly divergent lineages. Populations of different islands of Ternata brevignatha are genetically divergent, turning into what are possibly cryptic species. And what I think is the most interesting result, Ternata waikamoi is a widespread species overlapping with the other species on Haleakala volcano and without hybridization. Okay, so what this all means, if we have to kind of look back to our original question about the process of diversification. This all means that here we are providing evidence of sympatric distribution of closely related species of the same ecomorph, but without hybridization. This seems to be a snapshot of an intermediate stage where the species have formed due to isolation, they come together, but barriers for gene flow are already established. This co-occurrence of same ecomorph species seems to be less rare than originally predicted, and recent data on the annual radiation in the Caribbean and the cichlid on East Africa reveals similar situations. So how does that affect our original idea? We have this island with this single population, turns into 4 populations of different volcanoes, allopatry turned them into 4 species, and then we have events of secondary contact. This is the situation that we are illustrating with our genetic data. Secondary contact without hybridization. Now, what goes after that, and this is hypothetical, would be that this secondary contact turns into some kind of character displacement at an evolutionary level and makes this species differentiate in their ecology. And that could explain why we have different ecomorphs. But again, our data does not show this point, it really goes up to here, when we have secondary contact of same ecomorph species. So in a cartoonish way, this is what could be going on. From an ancestor, allopathic speciation creates 4 different species from the same ecomorph, but then evolutionary character displacement will create different ecomorphologies. But this is yet to be tested. So to finish, I would like to thank a large number of people who supported me during this work. This is part of my doctorate thesis already a few years ago. Many colleagues in the field in Hawaii, many collecting and protecting agencies, federal, state, and private, people working with the lab work and data analysis at UC Berkeley, and several sources of funding which allowed this to happen. Now, if you are interested in more detail, this work is already published in BMC Evolutionary Biology, and there's also a blog accompanying this publication that describes in a more broad language the main results. And with that, that's all what I wanted to tell you today. Thank you for your interest, and I will be very, very happy to answer any question that you might have.
Thank you so much, Darko. That was really very interesting and a very informative talk. It was a wonderful journey which you have taken us through. Any questions from the participants? If there are any questions, they are most welcome. You can also question me in the chat box, if at all. So I guess there are no questions. So thank you, Darko. Thank you once
Thank you.
again. And please stay tuned, and we'll have the pleasure of going through the work of Prasit as well. So please be with us to the national anthem. And now, dear friends, I would like to introduce you to our next speaker, who is also a very eminent person and a very dear friend of mine, Prasit Pongprom. Prasit is an ornithologist, an entomologist, and a nature educator. And he's from Rayong, Thailand. And the best part is that he is an assistant of Royal Forest Department, and he has worked on rare bird species in southern Thailand. In 2005 and 6, he has done as research assistant of National Science and Technology Development Agency. And he has also worked on spider diversity research in Western Thailand. Since 2008 to 2010, he was a lab manager with the entomology section at Queen's Curtis Botanical Garden. He was lecturer of biology and ecology at Science and Technology faculty at Nakhon Sawan Rajbhat University. In 2000, from 2011 till date, he's founded a Thai Nature Education Center at Rayong Province. If you go through his Facebook page, you will get to know all the wonderful activities he does with the kids, and I'm very impressed actually, Prasit, the way you carry on everything and the way you inculcate the love for the nature in the young minds, like in the students. It's really amazing. I've seen very small kids with you, so that's really a wonderful job you are doing. And Prasit is also working for Foundation of Rural Youth and Nature Education and Interpretation with Nature Education Foundation in the kindergarten as well. And he has started a learning center from 2012 to 2018. He was on the committee member and founder of Asian Society of Arachnology. So this is all I could tell about Prasit. So over to you, Prasit, waiting for your wonderful presentation. And if at all you have any difficulty sharing the screen, yes, Prasit is, so you can start. Just unmute yourself so that we can hear you. You're there, Prasit? Hello? Am I audible, Prasit? Prasit, we can see your screen, but we can't hear you.
Hello.
Yes. We can hear you. Yes.
Still. No.
Please carry on, Prasid. The screen is visible.
Wait a minute. What happened?
Yes.
You can hear me?
Yes, we can hear you. You're very much audible. Hmm.
गरम तो करून दे ना आता चालत नाही ना जास्त
तेच मी म्हटलं त्याच्याकडे आता असेल तर पाठवून दे म्हणा.
माइंड आहे म्हणून जास्त गरम नाही
मग देऊन दे म्हणायचं असतं ना आता. सरांना
What happened? Sorry. I will repeating and I sign again. Please wait a minute.
Okay.
Okay, but cannot share. Oh, disable. Host disable.
Not yet, not yet.
Okay. Sorry, my connection is signal is lost.
Okay, okay.
Okay, it's come.
somehow we had lost the connection with you, Prasid, but now it's okay. You can share the screen.
It's visible. It's visible.
It's visible.
It's work now?
Yes, yes, it's working absolutely
You
fine
can-
now. You can just start your talk, Prasid.
You can see?
Yes, yes, we can see as well as we can hear you, Prasid. You just start with it. It's working very well. Let's start.
No. Oh.
Yes, slides are.
You can see?
Yes, yes, Prasid, we can see you.
Okay, I can talk now.
Yes, yes, start talking.
Okay.
Okay.
Dr. Prasid.
Wait a minute. If what?
Yeah, yeah. If you're not using MacBook, then you can press F5. It will
F5.
start the slideshow.
Okay. F5. I don't know what happened in my
It's okay. It's okay.
okay,
There's
you
one
can
button at the bottom. There's one button at the bottom for the screen's slideshow.
yeah, but it's something is problem here.
There's few buttons there. The last one, the slideshow.
Yes, I know, but
It's
my
not.
bottom is close together. I cannot see the bottom.
Now it's active.
No, no, fix it. What
It
happened?
has been like right. Are you using MacBook?
No, I used HP
Oh,
Windows.
okay. Then the Windows, the platform is. Then you can press F5.
Oh, okay.
Yeah,
Now
it's
it's
right.
now
Yeah,
it's okay.
yeah.
Okay. Sorry.
No, no,
Uh,
it's okay, Prasid.
okay,
Going
sorry.
on. Yeah.
I would like to talk about spider in Thailand in general. Why we interesting of spider diversity in Thailand? Thailand is a small country, but rich biodiversity. So I work education with the children and work the research on spider together. Because in Thailand, we are no curator, researcher on spider at museum. In the Thai National History Museum, have a researcher on spider directly. Now in Thailand, we have only 5% work on spider and teacher together in for people in a university. And for me, I work on nature education with the children and general people in Thailand. So Thailand is very interesting the country. We have the population around 70 million people and have the 76 provinces. We have the parts of Thailand. We have 6 parts of Thailand and different part, different topography and climate. So why the Thailand have high biodiversity? Because about tectonic plates and lands evolutions. You see this is the place of Thailand and India together early Eocene and separate to Oligocene. And why spider from Thailand, some species different from India, and some species are different, similar from India, because we share together some it's a one time, including plants, animals, wildlife. Thailand's biogeography is divided to two main of the block, the place block, Shan Thai block and Indochina block. It's just two plates put together and try to combine ecological and species of plant and animal. Shan Thai is near Burma to India, and Indochina near China, Cambodia, Thailand, and Vietnam. And so in Thailand's effect on topography too, we divide topography in the north, in the mountainous area. We are highest of Thailand in the Doi Inthanon mountains, around 2,565 meters from sea level. It's the highest of Thailand. It's near Himalayas. We call Sino-Himalayan. And the Korat place to eastern Thailand, we call Indochina place. It's the Korat Plateau. This is the Jurassic land and different climate from another parts of Thailand, and similar to Laos, Vietnam, and Cambodia. And western is the mountainous area, and it's called Indo-Burmese regions. The plants similar to Burma and some in India, and the southern effect on by climate, rainfall, and animal. This plate, this region we call Sudai. You know, village line near. It is the narrowest of Thailand, near Prachuap Khiri Khan. And this is my hometown here, and this is Prachuap Khiri Khan here. This is divide to Thailand to pass the north and the south. Why Thailand now we try to protect the forest? The forest in Thailand, this is divide to tropical evergreen forest along the Thailand, almost near the south, near the equator, and scrub area, mixed deciduous forest, and dry littoral forest. You will see the most of the forest cover Thailand, almost in northern, western, and southern, but northeastern, not many covered by forest because the rainfall and effect of the place of the region. And now Thailand try to protect the forest by management. We call the forest complex management. We divide to the group of the forest and combine together for serve the biodiversity in 19 group of the forest in Thailand. This is the example of the forest in Thailand. If you come to Thailand, you find the dry diplocarp forest. In the dry season, the leaf fall off and very dry. And moist evergreen forest we found in southern and eastern Thailand. But only in the south of the Thailand, we have only one or two place of the peace swamp forest. It's small forest, but important for Thailand and for biodiversity conservation. And not in the highlands, we have the pine forest in the northeastern Thailand and northern Thailand. And the highest mountain, we have the mountain forest too. The most of the forest in the north of Thailand and northeastern of Thailand in the highest mountain. And near the sea, we have the mangrove forest along the coastline of the Thai Gulf. And we have mixed deciduous forest too, and cover in the upper of Thailand, northeastern, northern of Thailand. And along the beach, we have the beach forest along the south of Thailand, including Andaman Sea and Indochina Sea. Effect condition to provide the forest different and vegetation different, type of forest different, because we have the monsoon by southwest monsoon from Indian Ocean around May to October, and northeastern monsoon from November to April. So southwestern monsoon make the rain to Thailand, and northeastern make the dry and cold weather to Thailand, and some the middle of the years, the middle of the season, we have the typhoon, the storm affect by northeastern monsoon blow to Thailand too. So this is the rainfall in Thailand. We will see the southern top Thailand is the high rainfall in the southern and the eastern of Thailand. Now a status of the biodiversity in Thailand. You will see, but in Thai, but I have some word up in English. You notice insect and other invertebrate, but without spider. So spider like the new side of Thailand now, because Thailand just start to spider, especially Thai, I don't know, a spider researcher just have only 5 or 4 person who work on spider now, but around 40 years ago we have the foreigner from Europe come to Thailand, from America come to Thailand and done research on spider last around 40 years ago. But now the database of the spider in Thailand no have now. so so it's important because now the diversity, forest, environments in Thailand were destroyed by the people, but so very fast. But the rate of the new discover of the spider, but very slow. So I think it's it's important, and I worry about a situation in Thailand. But in my experience, I start to study spider in Thailand by biodiversity survey, because this is the basic to know how many species in Thailand, how important, where is habitat, what kind of habitat, and how to the conservation of the spider habitat, how to conservation, and how to tell people how to conservation and how important for ecological process in Thailand. So I have to work education together with the research together, because we have data of the spider now. Some group we have, but we lost spider because many people interested spider in Thailand, and I think the foreigner try to collect spider and try to sell outside Thailand. Spider of Thailand, we have the risk. I think the richest of the region in Southeast Asia. We have Leafetes, more than 33 species were described in Thailand, and many species are endemic spider of Thailand too, and some species disappear and no data after discovered by researcher. It is the example picture of some species of the Leafetes in Thailand, such as Leafetes aerowan, such as Leafetes ornatus, and Leafetes charugini. This is important for Thailand because we every mountain, every part of Thailand, you can find the new species, because the mountain separate together, the species are separate and different species too. But now the landslide, forest destroyed by people or villager, destroy the habitat of the Leafetes too. And spider group of the megalomorph now is the threatened species in Thailand too, because many pest market want to keep it and to sell outside of Thailand. In our list in Thailand, we have the Atypidi. Only 3 species were described, and recently we have the Halonoproctidae. Halonoproctidae were described by 6 species, and new family chain in Thailand. And Nemesidae, only 1 species. Nemesidae is very rare spider, and no data in Thailand. And Idiopidae and trapdoor spider are only 4 species. including Microstigmatides, one species, and Euclides, 8 species, and Theraphosidae, in my records, more than 13 species. So the true spider in Thailand, in my record, and I focus how the spider interesting in the big group of the Aranomorphes, such as the Araneides. Now, in my records, more than 50 species, but in the data that are cataloged by the World Spider Catalog, just only 2 species to describe a country list in that list. So the Araneides in Thailand is very interesting because in the diversity, including genus, general level, and species level, and many specimens and many species were not described and undescribed, such as the beautiful one, the unidentified spider, and now is unidentified and no described in Thailand. And Araneides genera such as the Parapecnata is no revised in Thailand. Araxnula, Irovisia, including a Gassaracantan. Last year, we have the news of PhD student. He revised the Gassaracantha in Thailand, proved the Gassaracantha and Macacantha diversity in Thailand. And Sotophora, this is a beautiful spider in Thailand and interesting, including the habitat, including the taxonomy level, including the described for new species. But in Thailand, not many researchers work on this. And South City very interesting tool, because just we have a list in the country list in the World Spider Catalog, only 21. But in my record and in my study, I found more than 40 species of the Southeast spider in Thailand. And this is the common Southeast in Thailand. It's very beautiful and interesting, including behavior, including the ecology, and including the effect of the habitat and the diversity of the Southeast City, the different habitat, a different group of the spider too, such as the Haemochilus, Cosmophasis, and Ticella, including ant mimic spider too. Tomicity for many countries describe the tomicity, but in Thailand now not have the researcher and arachnologists describe or revise spider family tomicity in Thailand. No revise. So in the world catalog, you will see the only 5 species were the country list in Thailand. But in my experience, more than 30 species and many specimens to describe to new of the spider in Thailand on tomicity. And Dinopidi, recently the Chinese Professor Su Xiangli described the Dinopis to change to the Asian Nopis. That is the image of the the one species of two species in Thailand in my collection, but in the list for Thailand now no have. This is a new family in Thailand, and we found this species, this family from the south to the north, and different shape, different species. Oxyopede, I think, is in India. You have this Fuchsia, and in Thailand we have two, but records in Thailand no have the archaeologists describe the Fuchsia and Oxyopede in Thailand too. So in Thailand we have a lot of species of the Oxyopede. So I think this family is interesting too. We described already by Dr. Ono from Japan. This we have only one species of the Eresidae in Thailand. We found from Western Thailand to Eastern Thailand. It is female and male. And Stenochelides, the ground spider in the live in the litter and the ground surface of the forest. This is important because in my experience we found only in the ground forest ground of the goose forest, but in the community, the people habitat, we cannot find the Stenochelidae. It's interesting, and now no have one describes Stenochelidae in Thailand. Corindidae is Deleman described many years ago, and we used the old data from Deleman. We have only 9 species in Thailand, but in my collection we have more in the and Tenidae too. No record from the world spider catalog, but in my collection more than 10 species in Thailand. So this family relate to the goose forest and dense forest, evergreen forest. So many topic to work on this Tenidae. Sparacity, the most of work from Peter Jaeger. He worked on the sparacity, especially heteropoda in Southeast Asia, including in Thailand, and one or two genera no describe, such as this picture. This is Telecopis. It's many new species and in Thailand, and Oleos. No spider researcher work on this too. Now we have just only 10 species record in Thailand. And Tetragonata interesting too, and there was the high diversity in the forest, and including in the agriculture area, including the chaff area and the right field. So you can see Tetragonata everywhere in Thailand, including the right field, including the forest. And we have more than 35 species, but in the report, world catalog, just only 3 species. So I think it's interesting. We have chance to study more and how to know the habitat and spider relate together. Cetodidae, This is the 6 species. The most of the work from Ono and from Chinese researcher too. It is important because we have one of the genus living only in the forest and cave, and one genus living near the people living area. So now we have only six species in Thailand. The last one, I think we have interesting the spider, the security. I think the around 10 years ago, arachnologists from Europe described secrus from Soviet Asia, including Burma, Thailand, and Laos. But Physinia lack the arachnologists describe on this genus. So, in my conclusion, why the diversity in Thailand, the spider diversity in Thailand, interesting, because no, we no have the researcher on spider, especially archaeologists, taxonomists, ecologists who work on the spider. So we need more for people, including the foreigner, including the Thai people too, to study more spider in Thailand. I estimate just 25% of the spider diversity we describe of all the spider should be found modern should be found in Thailand. Just only 25%. And spider evolution and biogeography, I think we need the researchers such as the Dago to work on evolution, because the Thailand we have ecoregion, try to get to 3 ecoregion, sub-region of the zoology sub-region, Indochina and Sunda and Burma and Himalayan, and endemic spider want to conservation now, because the rate of the destroying up to the nature the faster than new discoveries of the species and research. And finally, we hope to fulfill the spider database for Thailand by Thai people, by foreigner researcher from another country. We welcome. Thank you. Thank you very much.
Thank you so much, Prasid. Indeed, it was really a wonderful presentation, and we could see so many beautiful species in your country, which are, of course, some of them are common here also. I could find them here also in India. And as you said that Thailand is open for the researchers, and you are inviting researchers to work in your field with you. So we would be really very grateful if we could, as a Dr. Ambedkar College and Zoology Department, could collaborate with Thailand as well as with Germany and DACO and you specifically. So it was indeed a very wonderful presentation, and thank you so much. Both of you have made our day. So now I would request my colleague, Dr. DVNS Suresh sir, to present formal vote of thanks. And please stay tuned and be with us. Soon after the vote of thanks, we are going to raise for the national anthem. Over to you, Dr. T.P.
Thank you, Dr. Mishra. Ma'am, can you stop sharing of stop? Can you do the stop of sharing of his presentation? Because at his hand, it was a bit difficult. Yeah, thank you, ma'am. Thank you, Dr. Wong Prong, Dr. Prasid from Thailand. He's director of Thai Education Center, and the presentation was very nice. We had seen many species. Though I'm not from the field, though like I'm not an entomologist or a worker in on spiders, but I still these creatures, they have been made so creepy in our minds, because of these movies which have come up with these spiders concept. So every time I see a spider, I become a bit, it becomes a bit irritating for me. Sometimes I become very irritating also when I see some. But we have one faith in our India. Where the people say if a spider comes onto your clothes, you will get a new cloth in future. That was a myth. Maybe it doesn't have any relevance with the fact, but the relevance had something like, because it's a weaver insect. It weaves its nest, its web, and the clothes are usually weaved. So that way, maybe it's been linked with that myth. Anyways, thank you very much, Dr. Wong Prong, and Dr. Kotros is there. Dr. Taco Kotros. Yeah, Dr. Taco Kotros, right? First of all, I would like to congratulate Dr. Kotros for being the one of the fellow of prestigious fellowship, that's Alexander von Humboldt Fellowship of Germany, and the type of work he has been doing in Hawaii Islands, and the types of species, their evolutional background, the lineages. If at all he's there, may I ask, is there any lineage or linkage to the mainland spider species of US, the United States of America, with those Hawaiian species? If at all he's there, otherwise we'll move on.
There is a connection with the species from North America, yes. I don't know if you hear my answer.
We couldn't hear, sir.
Oh, can you hear me now?
Yeah, we can.
Yes, there is connection with species from North America for many groups of organisms, not only the spiders, but also with the species from the area of Russia and China. In some cases, hard to distinguish because at a higher taxonomic level, the faunas and floras of North America and Asia and China and Russia and China, for some groups, are very similar. So unless you have a very good sampling, it's hard to distinguish if it came from one place or the other. For others, it's easier, but for some groups, it's harder.
Thank you very much. Thank you very much. Actually, my wife is an entomologist, but her field of work is what do you call this? The beetles, Coleoptera. She worked on Coleoptera. Anyways, thank you, Dr. Kotros. It's been nice hearing you, and I again congratulate. That time it wasn't the connection was very poor. I couldn't hear you. On the behalf of our college and all the faculty from here, I would like to congratulate you for the. Sorry, there was a glitch. Thank you very much. And I would like to congratulate again for the prestigious fellowship you are enjoying in Germany and Sankenberg Museum, right? I think I have pronounced it right. Thank you very much, sir. We would be having future associations in days to come in any platform, or if at all you visit India to find some molecular lineages of your species and Indian species, if at all you extrapolate your work in that sense, we would like to meet you in India if at all you visit. Thank you very much.
That'll be great.
Thank you. Thank you very much, sir. And I would like also to, thank Dr. Prasit Wangprong. He is director, Thai Education Center, and he is a Thai Nature Education Center. That's a different type of institution they have. We don't have such kind of institutions in our country, but it's a bigger prospect institution, I suppose. They work on all the aspects of nature, and he has got a very good collection. I just wanted to inquire. We have heard about the spiders. The name. We have heard one name, one term called tarantula. What they belong to? What group or to what species or order they belong to? Like they have been like made so fearful in our minds. Like we always get fear. We are. I'm not getting the word to. we usually get scared by seeing those spiders. Yeah, the black widow, red widow. Dr. Prasid.
Thank you very much.
Yeah, okay. Thank you very much, Dr.
Thank you very much. Thank you.
On behalf of all the faculty, the Department of Zoology, and the Dr. Ambedkar College, Nagpur, India, I would like to thank you both, the speakers, Dr. Dako Kotaras and Dr. Prasit Wangprong, for sharing their collection, their views, and their study, their research with us on this day. Thank you very much for being the part of this meeting, this webinar. Thank you very much.
Thank you for the invitation.
Thank you very much. Thank you.
Yes. And now I take the opportunity to present a formal vote of thanks to all the organizing committee and different personalities giving different contributions to making this webinar a success. First of all, I would like to thank our President of the Param Pujya Dr. Babasaheb Ambedkar Smarak Samiti, Honorable Bhanteji, Honorable Bhanteji, Nagarjun Suresasai ji, for his actually he is a spiritual head in the community, and he has been the spiritual and the moral support for the college and the faculty all the time whenever we come up with some academic ventures and academic programs. Thank you very much, Bhanteji. And second, I would like to, our thanks are due to our secretary, Dr. Sudhir Fulchari sir, for his constant encouragement and support for all the types of programs we conduct time to time in different departments in our college. Thank you, Fulchari sir. And and then we move on to our beloved principal, Dr. B.A. Mehre. She has been a constant, a kind of inspiration, enthusiastic person in the college who keeps on pushing all the faculty to do something which is productive, something which is beneficial for the students, something which is beneficial to the research community as well. So thank you very much, madam, for being with us, for every proposal we come up with in front of you to get it approved. Thank you very much, ma'am. And then we would like to thank our organizing secretary, Dr. Archana Mishra. She's been she is the resource person in our college. She has so much of linkages and associations with the different associations all over the world, not only in India, all over the world. She has been a ardent worker in the field of spiders too. She works on spiders. She had interest in renal activities, also renal functionality, also renal research also. she had done her much of research in that too. And she has been in the nanosector of biological sciences, the nanophysics aspect of biological sciences. She's a good worker, and she keeps on visiting different. She's a going places lady of a college. Thank you very much, ma'am, for getting us such resource persons from different nations, for getting us abreast or getting us aware of the collection of spiders, the species spiders all over the world, so that if at all we have some interest in doing some research over the spiders, we can definitely get in touch with these people. Thank you very much, ma'am. And Dr. R.S. Bagre, being the head of the department, he has been always as a supporting pillar for any kind of work we undertake for academics or research or a student beneficiary. Thank you very much, sir. And I would also like to extend my thanks to the people who have helped us, though in the beginning of the session we had some glitch in the internet connectivity and all, but still we had thanks are due to Dr. Bongare from BCA department, who makes arrangements for all those those clips for the initial prayer, and the last we go for the national anthem, and his associates, and Miss Priyanka Jain, who had helped us, who has assisted in making the flyer, which we could circulate among the students and the different faculty. In a very short notice, we had to arrange all those things. Thank you very much. Thanks a lot for every contribution that each and every person who has made for making this success. Thank you very much. On the behalf of everyone and the Department of Zoology, Dr. Ambedkar College, I finally thank everyone for supporting and for contributing to make this webinar a success. Over to Dr. Mishra.
Thank you, Dr. DVNS. And my personal thanks goes to Darko, Kutaras, and Prasid. Thank you for being there with me. And may I now please request everyone to raise for the national anthem.
Sure.
तब शुभ आशीष मांगे, गाहे तब जय गाथा,
मंगलदायक जय हे भारत भाग्य दाता जय हे जय हे जय हे जय 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 हे
Sorry for the technical difficulties which we had. Was the national anthem audible to everyone? Hello? Dago?
Yes, I can hear you. Yes.
It was audible. The national anthem was audible.
Yes, yes, I heard it.
Okay, thank you so much. So on a good note, after the national anthem, I must say that thank you everyone. Have a wonderful day ahead, and let us pray together so that we get rid of the pandemic soon and get to know each other personally. And you would love the delicacies in India and waiting for you to be here in person. Thank you so much. Bye-bye. Have a good day.
Thank you for the invitation. Bye.
Bye. Bye. Bye, Prasid.
Bye bye. Thank you.
Joseph,
Thank you.
thank you, Joseph, for being there with us all the time.
Thank you.
Bye, Prasid.
Bye bye.
Bye. Bye-bye.
Bye bye.